We have no time to waste. So first thing on the agenda is grades must be fixed by tomorrow night. I will be here after school in enrichment today. I will be here at lunch tomorrow. If you want to start scheduling, I plan to be here every Tuesday at lunch. Okay, every Tuesday at lunch. Then you have tomorrow after school. So you have 3 chances to see me outside of class to help you with your grade. If you are in a serious situation, you can email me. Maybe we can set up an evening Zoom, which is hard because I don't have a whole lot of extra time, but we can talk about it. Or you can email me any work. I will be regrading everything tomorrow night to put in my grades on Wednesday. So maybe last minute, I'll do one more check after school Wednesday, but things need to be in tomorrow night. Remember our 16 and 17 syllabus statements. I know many of you are not happy with that grade. So we should be messaging each other, adding each other, fixing our stuff before I regrade those Tuesday night. Sorry I didn't change it on Google Classroom. You do not have another quiz today. You had your quiz Friday. Today we are finishing 18. Tomorrow we will start 19, and your test is Monday. I should have put that on here. Sorry. Your test is an 18, 19. Oh, it was on there. Your test is an 18, 19 hybrid test. Okay. So that will be on Monday. So we have this week to prepare you for a 2-unit test on Monday. It doesn't mean it's going to be extra long. Okay. It should be, we'll make it shorter than the last one. I know that was a doozy, but it will cover those 2 units. Your personal engagement should have been turned in yesterday. And data analysis part 1 is coming up. I think that's also 9/27. The original plan was for us to have already run some experiments. However, I know we are still needing to order materials and things like that. So tomorrow I will talk to you guys a little bit about when is my first order going out. If you're not done with your exploration and you don't know what we need to order yet, we need to figure that out. And I'm still on the bacteria thing. So I'll let you know when my first order will go out. For data analysis part 1, what I would like to see from you on that full paper post-here paper, I would like to see an empty data table that is organized for you to take your data when you run your experiment. Okay. So sometimes setting up a data table is difficult, especially with multiple trials and things like that. So I would like to see your attempt at setting up a data table for your experiment. And then I can look at it and give you feedback on is that good. Okay. So that's what I want you to work on. I know you haven't run your experiment and you don't have data, but you should be able to set up an empty data table. If you don't have a highlighter yet and you don't have one, you can get a box from the back. You're going to need that for your notes today. And I wanted to go through again the descent with modification idea with that whale example. So I'll give you guys a minute or 2 to discuss that at your table. How did our whale example go over descent with modification? Yes. Oh, syllabus statements. They should be due when we're done with the notes, right? So yeah, yes. So this should be due today. Sorry, you guys, sometimes if I change the schedule, I don't always remember to go change it in Google Classroom, and I should, and I apologize for that. Your syllabus statements are due today. Your notes technically will be due today. However, in terms of collecting your notes, I shouldn't collect your notes when you have a test coming up, right? So your notes should be done today after class. I will collect them on the day of your test, which is Monday. And your unit 18 quiz was Friday. Okay, so everything's all up to date. Okay. I don't hear us talking about the whale example. Whales, how did that show us a descent with modification? Through the fossils, okay? Start there. Through the fossils, we saw Certain traits kind of slowly changing over time, right? What were some of those traits? The legs, yeah, the legs, the back legs basically shrank and went away over time. The nostrils went to the head. They actually merged into one big nostril on the head. The eyes? Oh, the location of them? Yeah. So I think that was another thing too, the location of the eyes from the side of the face to the, no, from the front to the side. Yeah, because when they were land-dwelling mammals, not mammals, land-dwelling mammals, they were more forward seeing. And then it went to the side. The size, yes, that was another thing. They got bigger. Something about the feet. They went from hooves to webbed, from one hoof to two hooves actually, and then to multiple digits. Okay. We talked about the hind legs. Good. So those were kind of the fossil evidence that showed us this idea of descent with modification. That as these ancestors reproduced and these different offspring over generations gave us different species over time. So descent, because that is talking about multiple generations of time. With modification, those modifications, what causes the modification? Mutations. So all the new traits were caused by random mutations, but those mutations were what? Causing them to survive. Thinking a B word here. Beneficial. Those mutations were beneficial for the organism. So because they were beneficial over time, the organisms with the beneficial traits survived and reproduced, and the other ones died off. And then that changed the frequency of what in the population? The alleles. Very good. So remember that was some of your 5 tenets of evolution, of natural selection. Okay. This is where we are in the notes. You have this. I wanted to kind of go through and highlight parts of this. I believe this is tied to one of your syllabus statements. These scientists, what are their names? Read it. Peter Parker and Rosemary Grant. This is part of 5.2A1 syllabus statement on page 2. It says, to outline the role of Charles Darwin, okay, we all know Charles Darwin, and Peter and Rosemary Grant in the study of Galapagos finches. So you do need to know these 2 scientists. So please highlight their names. So as you are studying your packet, you will remember, I should know these people. Okay. So these people basically kind of kept Darwin's observations and not really experiment, but his observations of the finches going. So on the Galapagos Island, the finches. Okay, if you don't know the finches, I'm hoping you learned that freshman year. But basically, Darwin looked at the beak sizes of the finches. Okay, and what he noticed, I should put, sorry. What he noticed is that different finches had different beaks, right? So if I just look up Darwin finches, there's so much that you could read about and look at. So what's a good image? This is a good one. Are you blurry? No. Okay, so Darwin and his finches. Darwin noticed a lot of things, right? He noticed different traits in similar like species in all these different types of things. He traveled all around South America and observed. But his most famous example is the finches. These are all finches. As you can see, their beak size and shape is different. Specifically, if we look at the large ground finch, it's very wide, kind of short, versus the warbler, very long and skinny, okay, and everything in between. And what he noticed is it correlates to what they eat, okay? So if we look at these kind of wide, thick beaks, those guys were seed eaters. If we look at the warbler and some of these other kind of long and skinny shapes, they were insect eaters. And then we've got this vegetarian fruit eater over here. So in terms of different traits, again, if we think about our tenets, we have variation in a population. And for these guys over here, it was beneficial to have this shape of beak because they have to crack open seeds. So their beak has to be super strong. Okay. This guy, warbler, probably not very efficient at cracking open a seed, at least not big ones, right? But if you think about being an insect eater, you have to be able to get into somewhere and pick out the insects. So it might be smarter to have a small skinny beak. Okay. So that's Darwin and his finches. He noticed these differences and he figured it probably ended up that way through descent with modification. Okay. So Peter and Rosemary, along with other people, have been carefully documenting the ups and downs of the finches on the Galapagos island of Daphne Major. Even in this relatively short amount of time, they have directly observed the evolution of body size, and beak shape in response to changing ecological conditions, such as droughts and El Nino events, both of which would alter the amount, the sorts of seeds and the food available, and the invasion of other finch species that compete with locals for food. So if we think about our 5 tenets, these guys for over 30 years have been watching basically kind of evolution happen. Okay, so in terms of highlighting key points, what do we think would be important to highlight other than this? Let's do some close reading. Okay, good. I would say evolution of body size, beak shape. That would be good. So who are they? What are they looking at? Okay. Yes. And in response to changing ecological conditions. I would stop there. Yes, El Nino is one of those. Droughts is one of those. You could probably think of other examples, right? But we just want the key information. Good. I agree with that. Also, invasion of another finch species, because that's not really I guess it would be an ecological condition in a way, yeah. So yeah, so basically you want to know that Peter and Rosemary kind of continued what Darwin did. Darwin was kind of different. He moved around to a lot of places and he noticed, okay, there's birds over here and birds over there, and they're similar like this, but they're different in these ways. So he was kind of focused on the differences. These guys stayed in one place and they focused on what was happening with these Darwin's finches over time. And as you can see in the graphs, beak shape over time has changed. And this specific species of Geospiza fortis. So if we want to analyze this graph, I should put this way, people can hear me better. The beak shape went from blunt to kind of more pointed, back to a more blunt shape, back to a more pointed shape, and then kind of evened off. And that we wouldn't need to know more about what was going on, but this dip right here is specifically interesting, right? What happened to the food in this case, in this span of time, that caused those beaks to change shape? And then over here, we've got a different species, Geospiza scandens. Same span of time, 1970 to 2000. Their beak shape started out pointed and went more to a blunt shape. Okay, so without a lot of information about exactly the ecological changes, we can at least see that they are seeing these differences. Okay. Our next example, which I'm not going to spend too much time on because we have a whole section in our notes, is antibiotic resistance. These examples are important because natural selection and evolution, like in the video, we talk about how this happened over a long period of time, right? It took a long time for us to get all these different organisms. But these are some things that we can literally see, like Peter and Rosemary from 1970 to now, we see evolution happening. Currently, we can see evolution happening in antibiotics because they're building a resistance. So let me read it to you and we'll think, what should we highlight? Antibiotics are drugs that kill or prevent the growth of bacteria. When antibiotics were first discovered, they seemed to represent a miracle cure for human diseases like pneumonia, typhoid, bubonic plague, and gonorrhea. However, almost immediately after the introduction of antibiotics, bacteria began to up the stakes. Resistant strains of bacteria soon evolved that could grow even in the presence of and I cut that off. You have it in your notes. Thank you. Perfect. Okay. So what's important to highlight from here? Not everything. Okay, so I would say antibiotics kill or prevent bacteria. Antibiotics kill or prevent bacteria. We need to know what an antibiotic is, right? Actually, you learned that in Unit 3, way a long time ago. Okay. This kind of stuff I think would be kind of too specific. We know that they were good for a while, right? So jumping down to kind of the bottom part, what else should we highlight? Yes, resistant strains of bacteria soon evolved. Resistant strains of bacteria soon evolved. And then maybe rendering our drugs ineffective. Yes, and like I said, we're going to go more over this. You do want to know that graph, though. Notice that graph. This graph is showing the resistance to the antibiotic vancomycin rose dramatically over the 1990s in U.S. hospital intensive care units. So 1983, 1989, it's good. Vancomycin was working, and then all of a sudden it started evolving and becoming resistant. Then what do we do? Oh man.
Natural selection.
Yes, Sandra, I kind of heard what you're saying. Okay. Our next example, industrial melanism. We could probably talk all day about this example. This is another pretty recent, less recent, but still pretty recent case study of natural selection evolution. We're looking in the late 1800s, and we even see some data from kind of recently, late 90s. I was like a little girl in 1998. So industrial melanism, you want to know that this is connected to moths, as you can see, moths. The frequency of dark forms of the moth increased dramatically as coal burning in England blackened the surrounding landscape and allowed dark moths to survive better than light forms. So what was happening, which you might want to add like to this little section somewhere? What history buffs? Late 1800s, cities like England, industrial revolution was happening. So this is an environmental change caused by humans, which we are often the cause of environmental change, right? So in the beginning, let's look at the frequencies. In 1848, pre-industrial revolution, there were trees and their bark was nice light colored bark, light gray, whitish colored bark. So what survived better? The light colored moths. There were dark colored moths because what was happening in the population? The population had what? Genetic variation caused by a mutation. So then industrial revolution happened. Thank you, humans. And all of the trees turned black because of all of the soot we were polluting the air with. So no longer do the white moths survive. Now the black moths are surviving. Would the white moths survive better?
Because there's no
Blend in, camouflage, they don't get eaten. Very good. Now, if we scoot forward to 1998, what did we see happen to the frequencies? They reverted back because Industrial Revolution, we did not care at all about pollution. We had like no thoughts on, maybe some people did, on what it was going to do to our environment. Then we went through a period of time where like, oh, this is killing people, lung disease, like bad things are happening. We still pollute, right? But we made a lot of advancements in 1998. My uncle was telling me one time that he was like a teenager in like the 70s. And he said, you could never see LA, like driving on the freeway. There was so much pollution. Like everything was just brown always in the 70s. So we think pollution is bad now, but it actually used to be way worse. Yeah, and a lot of it is invisible pollution. Okay, so what are we highlighting? Frequency of dark forms increased during Industrial Revolution, something like that. Okay. Frequency of dark forms of the moth increased during, I would add Industrial Revolution, something like that. The rest of it, I think you guys could probably figure out why. Like you guys understand, right? With recent increased pollution restrictions, light moths are once again favored. So maybe something like that too. I would actually, on this one, mostly what I would do is I would highlight the frequencies to show how they've changed over time. Okay, and the dates, yeah. And definitely, if you haven't written Industrial Revolution on there anywhere, there's actually a graph. I should find it and I should put it in here. It shows a bunch of pie charts. And what they noticed, not only like, basically, this data was taken in England. England was a major city, right, where a lot of Industrial Revolution was happening. What they also noticed was that at the same time that England was having this increase of dark moths, if you looked at the countrysides, like way out in the country where there was no pollution, they did not have a change in dark moths. Way out in the countryside, they still had more light moths on their trees because their trees didn't turn black. So it's just kind of added evidence to this idea of natural selection that it only happens when the environment changes and when you have a variation in the population. Okay, so you have three really good examples of natural selection that you're going to want to be familiar with. Anyone want to guess the #1 most frequently asked question of IB biology exams of all time?
How do you answer for questions?
No, like questions that you will be asked.
Oh, frequency.
I'll just give it to you. Explain the sources of biological variation in the population. This might be a multiple choice. This might be a database question. This might be just a straight written response. And if you don't know this, that would be silly because it's literally on almost every single test every single year. It's not in your notes. I know this is like review. We have a whole section of notes. Sources of, yeah, page 5 and 6. Page 5 and 6 is the information that this is asking for. Okay. This is just saying you're going to find a question like that covering this topic on a test. Okay, let's do this matching. I'll give you guys a couple minutes to figure it out. And I'm going to call on cards. Yep, start with the obvious and go from there. If you can't see B, B is a picture of metaphase. And A is a picture of little spermies. I don't know.
Vice President.
I showed my freshman a video of the journey of the sperm from the vagina to the egg. It was actually a very good animation. They were actually really good, maybe more mature than you guys.
Hey, you didn't have
Oh no, I'm sorry. I lied. It was actually my junior. They were good at that. Are we ready? Angel, pick a number and tell me the answer. What'd you say? E. Oh, we didn't do our would you rather. Lame. Okay. 3 is E. Do you agree with that, Nate? You don't? What do you think? Okay. I was like, that was the easy one, guys. Ricardo, another number. 4. A. I'll go with that. Very good. Jessime. 5. 5C. Giselle, do you agree? Yes. Very good. All right. Jesus.
Five. Oh shoot.
One or two? One is D. What do you think, Jaden?
Yeah.
Make sense? Okay.
Last one.
Lucas. 2 is. You're gonna say B. I'll go with that. Very good. But why? I should have asked you guys why. Yes. Independent assortment is kind of like it sets up in metaphase, but anaphase pulls them apart in different combinations. So basically, independent assortment, because the way they align in metaphase can align in multiple different ways. Anybody else need an explanation of why the answer was correct? Okay.
Yes,
Good.
you rather. Oh, we already answered that.
Oh yeah. No, I know.
We need a different one.
You want to pick it?
Yeah. Kessler, no, wait, Kessler, it's his birthday.
It's your birthday.
Uh,
Happy birthday to you. Happy birthday to you. Awesome.
Number 6.
Go ahead. You can pick a different one. It's your birthday. Pick whatever one you want. Okay. We're gonna skip this. I do want to go over this. Calling my game whack. Okay, let me know when you find one. I want you guys to discuss these answers at your table. I'm calling on more cards and I'm shuffling, so you may get called on again. Maybe Jaden will have the magic touch. We're not making up our own would you rather. Let's do this until we find one. Our industrial melanism, which is the moths, answer these questions. I may call on you. You have 30 seconds to choose one. This one?
That's a solid one.
Oh, sorry, first period. Would you rather?
Oh,
Okay.
wait, what was the slogan?
All righty. I hope you're ready. Kalia, what was the variation in this example? What was the genetic variation? What were the different options?
Pick from the bottom.
Black and white moths. Good. Black and white moths. So we could maybe make that more fancy by saying the amount of melanin in the wings of the moth, something like that. What was the selective pressure, Daniel?
Pick from the bottom.
Yes. More noticeable to who? Predators. Good. So that was the selective pressure. The pressure was predators. Okay. The change of the environment caused the black to survive better than the white, but the selective pressure is always the same. Are they going to be eaten? Okay. Good. Yes.
So I guess that makes sense. Like I thought selective pressure could also be like environment.
It could be if that change is threatening the species. In this case, the pollution wasn't killing the moth. So in terms of like if it's a drought and you can't get enough water, then that would be the selective pressure. Yeah. In this case, they didn't really care about the pollution, but it's the predators that was the pressure. And then the change in pollution changed who survived better. Who was able to survive and reproduce better? Ricardo? The black moths, very good. How did the population change as a result, Patricia? What happened with the frequency of the alleles of the black moth? Good. So the black moth frequency increased, the white moth increased frequency decreased. Okay, cool. Moving on.
Oh, yeah, the butcher.
Okay, this is more review. Go ahead. For each of the 5 components, we call them tenets, those 5 parts of natural selection. What are we seeing here? First, what are our 5? Let's see if we can do this without looking at our notes. What was number 1?
7. Overpopulation.
Overpopulation. Number 2. Variation. Number 3.
Pressure.
Selective pressure. Variation kind of goes with mutation.
Difference in frequency?
That's number 5. A change in frequencies.
Yeah. Or survival.
Or is like natural selection.
Yep. I think it stated differential survival. Survival, but yes, natural selection. Okay, which one are we seeing in this picture? Well, this is 1976. This is 1978. I would say, yeah. Yes, I would say this is more giving you data on the frequency. Yeah, which is caused by number 4. What's happening? We're looking at babies.
Oh, this is nasty, bro.
Which of the 5?
Overpopulation.
I don't know what overpopulation that makes sense. Angel, are you sleeping? I'll be sleeping.
Oh.
What is going on?
Stop making these Yo.
Reproduction will probably go with
In the air, though?
Oh,
Overpopulation.
yeah.
Run for a running start.
Dragonflies, they do it in there.
Man. I just got a new level of respect for dragons.
They do, and they make this like anyway.
I couldn't explain it.
Like what birds do they like spiral down and then at the last minute they go like that?
More than one.
Like eagles.
Yeah, when they fight, and sometimes their claws get stuck, and then they can't get unstuck, and that's really scary. Okay, what are we seeing here? Abundance of seeds,
So good.
and we're seeing we're looking at, guys, you need to know how to read graphs. So what are we looking at here? We're looking at over time
Abundance of seeds.
and the abundance of seeds. So what's happening with the abundance of seeds when I do this? Growing. It's crashing. What does that match with? Selective pressure. Very good. So the food is going away. That's a selective pressure. Yes.
I was wondering if that's a change. It depends on like, right? But if we're looking at it from like the seeds point of view, it's kind of
Yeah, I guess you could actually say it's a change in frequency too, because, well, not really, because it's not really showing you that there's a variation of seeds. It's just saying this one seed is there or not there. So yeah, not really. Okay, here we go. Antibiotic resistance. You are on page 10. You shall trace this graph. On the bottom, put year. You don't have to write all the years. Just put time or year. And then percent on the top. You do not have to write out each percent. You want to know the trend. That antibiotic resistance has this kind of trend. Ooh, from math class, what does this match? It's exponential. It's exponential maybe if I stop here-ish, but not really. Exponential would go up a lot faster. I would call it logarithmic.
Linear correlation.
It goes up and then kind of evens off. Okay, it increases and then evens off. This is a very common graph that we see with populations. Populations that are new will increase because there's plenty of resources. And then at some point, they increase so much, there's not a lot of resources, and then they kind of level off. Okay? You don't have to draw the dots, just draw a line. Just draw a line that goes flat in the beginning, up, and then starting to level off. You have about 20 seconds to get this graph written. Thank you. Moving forward. Let's notice some other graphs here. This one is showing you increase in antibiotic resistance of 3 specific infections. MRSA, if you have anybody in the nursing or doctor or hospital field, you ask them about MRSA, they will tell you it's bad. When that shows up in hospitals, we are scared because we don't have anything to fight against it, really. I forget what it stands for, but it's a specific type of bacterial infection that we currently don't have a lot of great antibiotics for it. And it spreads really pretty easy. I don't know what these two stand for, but each color is a different type of bacterial infection. And the graph is showing you the resistance. So look at MRSA. MRSA is really up here. It's scary. But in all the cases, we're seeing that they increase in resistance. We are seeing a similar trend in this one. Over time, the number of enzymes identified that are antibiotic resistant enzymes has increased. It's bad. It's a specifically gonorrhea, a sexually transmitted infection, has been developing resistance to the antibiotic we used to use. So we developed an antibiotic in 2006. There's a lot of chatting. So in 2006, we developed this antibiotic. It worked for a while. And then gonorrhea was like, nope, I'm going to evolve. And now your antibiotics don't work anymore. Probably working. This one I thought was interesting. Antibiotic use. We're going to go over how can we stop this resistance? Well, it has to do with overuse and usage. So if we look at different countries, this on the bottom is the outpatient use of penicillin. Penicillin is one of our first antibiotics, right? Daniel, you should not be typing. What was that? Penicillin was the first one, right? Remember Alexander Fleming, a long time ago, developed penicillin. This is the proportion of resistant bacteria. So what happens as countries use more penicillin? What happens to the resistance? It increases, right? So France uses a lot of penicillin. And they also have the most proportion of resistant bacteria. But if we look at Germany, for example, they use penicillin a lot less. Therefore, they have less proportion of antibiotic resistant bacteria. Are we on this graph?
United Kingdom.
No. Oh, that's Europe. Sorry, we're not in Europe. Duh. Sorry. I think we'd be more up here though. Okay, so trends observed. You have a bubble. 1, 2, 3. Number 1. When new antibiotics are used, resistance will eventually establish. So in this picture, what does the big scary green guy represent? The bacteria, yes, which we can say germs for bacteria and viruses. So bacteria. And the poor scared little red and white guy, that's our antibiotic. Viruses are not affected by antibiotics. That's why if you get the flu, you should not be taking antibiotics because it's not going to do anything for you other than kill your gut biome, because your gut biome is good bacteria. We'll get into that. So over time, since antibiotics have been used, there have been changes in the resistance. Okay, so we see that resistance eventually happens. So kind of preluding, we need to talk about how can we stop it, right? If we know this is a problem, humans always try to fix problems. How can we fix the problem? We'll kind of get into that. Here is something we don't have time to read the whole thing to, but this is a new antibiotic. The first member of a new class of antibiotics, and more importantly, the method used to discover it could lead to many more. So just kind of the idea that, like, as humans, really smart humans, we notice there's a problem, we need to figure out the answer, right? So apparently they've developed this new kind of antibiotic. Number 2, resistance has spread to new species of bacteria. You're writing number 2. Number two. So what we know about bacteria is they can actually share DNA. They come close together and they have something called conjugation, and they can send bacteria from one bacteria to the next, therefore spreading their resistance abilities. This is a petri dish. This is a common way where we test to see what antibiotics are functional for what bacteria. So this would be a bacteria growing, and each dot has a different antibiotic on it. And as you can see, TE30 is very effective. S10 is very effective. P10, not so much. That's just kind of a way in a lab we could see which antibiotic is going to work against this bacteria. They do that test. The TE? Bottom left?
Yeah.
This one?
One circle.
Oh, yeah. Yeah, so it's like it's really effective, but then the effectiveness kind of starts to wane. Yeah, that's definitely the difference in patterns. I don't know exactly what that means, but possibly you are right about that. Number 3, the proportion of infections caused by resistant strains have increased over time. Yes. This one might be the control, I would assume, because it doesn't have a number. So that probably just has nothing on it. So they can see that the tab itself isn't affecting it. What do you mean in terms of like our medicine?
Yeah.
I don't think so. Because whether something's going to fight an infection or not has nothing to do with what we feel or think about it. Yeah. This is just a 2019 piece of data. Bacteria and fungi listed in the 2019 AR threats report. Hello. We've got, we don't need to necessarily read them all, but look, drug resistant gonorrhea is one of them. But as you can see, we've got some urgent threats and serious threats happening because of resistance. We're supposed to review the 5 tenets. We just did that a minute ago, so I'm going to skip that. I'm sick in my head all day. Okay. So remember our 5 tenets, they're on the board. I'm going to skip this. Explain how antibiotics evolve resistance. So basically, we want to apply these 5 tenets to specifically evolution of bacteria. So number 1, you're writing in the description side. There is overproduction of bacteria in the population. You could just put overproduction of bacteria. You want to shorten that. And then in the sketch portion, you're just going to draw yourself a bunch of little bacteria. You don't have to draw that many. I would say draw at least 5. You finished? Gotcha. No, it's good. Good. I forgot this. I have it in my hands. All right, so first, overproduction. Alrighty. Number 2, bacteria have variation in their susceptibility to an antibiotic. You don't want to just write variation. You want to write what is the variation. In your next step, you're going to draw a bunch of bacteria and color one of them. Whatever. But the variation is in their susceptibility to an antibiotic. In the beginning, all the bacteria would be killed by a certain antibiotic. Poor Alexander Fleming would probably be sad to know that his penicillin is not very useful anymore. It is still useful. There are many things that it will not save us from anymore. But he would probably still be happy to know that he started this trend of antibiotics. All right, so our bacteria have variation in their susceptibility. So that little yellow bacteria is like, I am mutated, I am resistant. I will survive. I will survive. All right, 10 seconds. Or less. Oh no, we're mostly done. Okay. Number 3, selective pressure. The antibiotic is the selective pressure. You can even just put antibiotic equals selective pressure. And it will kill the bacteria that are not resistant. So in this picture, draw a little bacteria again and X out almost all of the ones, but not the one you colored. You can leave the extras because in realistic terms, when we take antibiotics, it might not get every single organism, but it'll get the majority and then our body might take care of the rest. Because our body can fight infection on its own. If your infection is not a bad infection, your body can take care of it. You get something like MRSA, your body can't handle it. Or if you're immunocompromised, something like that, your body cannot handle it. Tetanus, is that a virus? It's a virus. That's why you need a vaccine. I actually recently got my Tdap vaccine because when you are pregnant, you get your Tdap vaccine because it will protect your growing baby. Tdap is tetanus, diphtheria, and pertussis. Pertussis was the one that I feel like they mostly give it to you for because pertussis is whooping cough. And whooping cough kills babies. Baby probably isn't going to get tetanus anytime soon, but you never know. Okay, next. There is different. I know that. That is a creepy, creepy cartoon. There is differential survival and reproduction. I'm glad it still lives on. Rusty spoons. Orlando doesn't know about rusty spoons? Yes. Oh my God. There is differential survival and reproduction. Basically, the resistant bacteria live and the non-resistant die. You can, I'm sure, shorten that. But in this sketch, you're going to draw some binary fission of your resistant bacteria. Okay, remember differential survival and reproduction. It's nice to survive, but evolution isn't going to happen unless you actually reproduce and pass on those new traits. Oh, Elmo always creeps me out.
What about Cookie Monster?
I think it was when Tickle Me Elmo came out, and I didn't feel great about it. I don't know. It just gave me like a creepy vibe. I was scared. I wasn't really when I was like 2. All right, do we have our binary fission?
Yes.
We got to finish this. We got to get one more box before we leave today. Yeah, it's weird. Number 5, over generations. The frequency has changed. Number 5, on our tenets of evolution. Frequency of resistant trait increases in the population. So now all your bacteria should be colored, whatever color. I had a similar experience. When I grew up, there was a place that was not quite Chuck E. Cheese, but they had like it was Rocky and Bullwinkle. I don't know if anybody knows Rocky.
Oh yeah, I know that.
Okay. So it was like a Rocky and Bullwinkle place, and I remember they came up to me and like tried to give me a hug, and I yeah, no, my mom was like,
We were at SeaWorld, my sister did not like him, Susie.
yeah.
The yellow or the sign?
Okay, as you guys are finishing up, your would you rather of the day is, would you rather be friend zoned by your crush or have your best friend fall in love with you? Friend zoned by your crush or have your best friend fall in love with you? I had a situation where my best friend confessed his love and it was awkward because I did not feel the same. I was like, let's just pretend that never was said and go back to how things work. And we're still friends actually, so it's all good. But it was weird for a minute. All right, you guys, test on Monday. I know the syllabus statements are supposed to be due today. We didn't finish, so you guys just know syllabus statements should be done by the time we finish the notes. Okay? And don't forget to fix your old ones. Bye. Make sure you are messaging each other, being supportive to each other. Hold on a second.